because actually I'm now on leave actually. I have a meeting with someone. So actually my meeting was scheduled at 12 a.m. However, the person that I uh, need to meet change the schedules. So saya dah jumpa dia awal sikit. So saya terpaksa buat waktu petang, waktu lepas lunch. Biasanya saya tak, I'm not really like to have class after lunch. Nanti sebab student rasa mengantuk sikit. Awak mengantuk ke? Awak okey ke? Cergas ke? Mengantuk ke? Okey semua?
Okay.
Okay.
Okay. So before we continue with our topic for today, I would like to remind you that this week it is our third week of semester. Ni minggu kita yang ketiga kan. So in our course planning, kalau awak boleh rujuk balik dalam e-taklim minggu ketiga, you will have a quiz. Tapi saya tak upload lagi. Nanti saya upload soalan tu and then saya bagi masa selama 1 minggu. Okay. So starting later if I have upload the questions, I will inform you. And I will give you a week, very ample time for you to answer the questions. Okay. But doesn't mean you have a week for answer the questions. Once you click the questions, you only have 30 minutes. The questions, only 2 questions, okay, from first lecture. Okay. And then don't forget, next week we will have face-to-face lab sessions. Okay. 2:30 o'clock at lab, dekat lab plant science apa nanti saya confirmkan balik, tapi dekat physics building. Okay. So that will be our face-to-face. For class, we will still conduct our class on secara online, because untuk buat masa sekarang only lab sessions and also FYP yang boleh face-to-face. Okay. So before we continue, I forget, sepatutnya saya sambung lecture minggu lepas kan, ada beberapa slide yang tak habis kan. And some of you have done the tutorial questions that I upload in e-taklims, okay? But I'm sorry, saya tak sempat nak go through. Siapa yang dah hantar tutorial yang buat tu, saya akan go through nanti. Okay. So without further ado, let us continue to our lecture for today. Okay. So before that, let us recite Al-Fatihah dan baca doa. Bismillahirrahmanirrahim. Alhamdulillah. Bismillahirrahmanirrahim. Allahumma ftalaina hikmataka wa anshur alaina min zaini rahmatika ya arhamar rahimin. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Okay. Can you all see my slide?
Yes.
Yes.
Okay, last week we have discussed about the elements that we required by plant for their nutrition. Okay, out of 180 available element in our nature, plant can absorb nearly 60 different kind of elements. Okay, however, if we cross-check between different species of plant, we will observe that all species of plant require very specific elements. There are 17 elements that when we observe in all different species of plant must have this kind of elements. Okay, so these elements we call as essential plant nutritions, okay, or essential elements required for plant nutritions. Okay, so all these 17 elements share 4 distinctive characteristics. Okay, So I hope you can remember the characteristic of essential elements. Um, Generally, these elements can be grouped into different group based on their sources. For example, if this element we classify from based on their sources, we can classify all these 70 elements to different groups, which is minerals, which is the elements uh, absorbed by plants from soils, and also non-minerals, which are the elements that acquired by plant from atmosphere and also water. Um, we can also classify the 17 elements by the amount required by plant. If we classify all the 17 elements based on the amount required by plants, we can classify this element into macronutrients and also micronutrients. So macro mean required by plant in large amount, but we must uh, draw a definite line how much plant require this element so that we can classify these elements as a macronutrients. Berapa dia punya concentrations in plant dry mass, if you still remember? Ingat tak? Um, 96% is if we do chemical analysis, carbon, hydrogen, and oxygens comprise 96% of plant total biomass. But for macronutrients, um, the amount of these elements must exceed 0.1% of total plant dry mass. Okay, if you do chemical analysis, you determines how many percentage, how much a certain elements contained in your plant samples. If the elements, the amount of the elements exceed 0.1%, we call the element is macronutrients. And we have done, the scientists have done the chemical analysis and found that uh, um, uh, 7 elements, excluding carbon, hydrogen, and oxygens, a group as a macronutrients. They are nitrogen, phosphorus, potassiums. So these 3 elements we call primary macronutrients. Okay, micronutrients can be further divided into 2: primary macronutrients and also secondary macronutrients. Okay, so macronutrients are elements that contains more than 0.1% of total plant biomass. So these macronutrients, like I said earlier, can be further divided into 2 groups: primary macronutrients and secondary macronutrients. Again, we must have a very clear line to differentiate between primary macronutrients and secondary macronutrients. So what is the difference between primary and secondary macronutrients? Their amounts. Primary macronutrients required in very large amount compared to secondary macronutrients. But how much that element required by plants? How much the difference between primary macronutrients and secondary macronutrients? For um, primary macronutrients, if we do a chemical analysis, the total of primary macronutrients in from total plant biomass is more than 0.5%. Okay, kalau kandungan elements tu 0.5%, lebih daripada 0.5%, kita panggil dia primary macronutrients. Okay, and that elements are nitrogen, phosphorus, and potassium. Okay, so in plant nutrition, usually we don't discuss about carbon, hydrogen, and oxygens so much, because these 3 elements are abundantly available in our environments. We always discuss about mineral elements, elements that being absorbed by plant from soils. Okay. Um, tadi saya cakap macronutrients, primary macronutrients, dia punya kandungan dalam tisu tumbuhan adalah lebih besar daripada 0.5%. Okay. Untuk secondary macronutrients, calcium, magnesium, sulfur, tiga ni lah, okay, magnesium, sulfur dengan magnesium, calcium, sulfur, magnesium. The concentrations of these three elements is range between 0.1% to 0.4%. Okay, so remember macronutrients overall, generally macronutrients, their concentrations in plant biomass is more than 0.1%. Okay, 0.1% and higher, we call the elements macronutrients. We can further divide this element to primary macronutrients and secondary macronutrients. Kalau primary, dia punya kandungan 0.5% ke atas. Kalau secondary, dia punya kandungan 0.4 hingga 0.1. Okay. So, and then the rest is micronutrients. Okay, so macronutrients ada berapa ya? Saya tak ingat lah. Kita ada 6 tambah 3, 9. So 8 adalah micronutrients. Okay. There are 4 characteristics of this essential element. The last one is the depletions. Okay, if plant experience deficiency of these elements, plant will show specific symptoms. Okay, so how we can analyze, how we can know that certain plant experience nutrients deficiency? Bagaimana? Pokok dia akan tunjukkan symptoms. There are different kind of symptoms of nutrients deficiency that we can observe on plants. Okay. Basically, the diagnosis of these deficiency symptoms kita boleh bagikan kepada visual diagnosis. Okay, we just determines what kind of element involved by visuals. Just observe the symptoms. Okay, there are certain way how to determine what kind of element involved in plant nutrients deficiency. However, visual deficiency, even though it is very fast, cheaper, but it sometimes can be inaccurate. Okay, visual deficiency ni paling senang, paling murah, tapi biasanya kita boleh misdiagnose. Okay, dia tak begitu tepat. Especially kalau nutrients tu dia mempunyai fungsi yang hampir sama in plant physiology. Okay, for example, magnesiums, nitrogens, ions, they are all required for chlorophyll synthesis. So the deficiency of this element will show chlorosis. Okay, so we can sometimes misdiagnose. Okay, kita tersalah diagnose. Okay, kita anggap mungkin magnesium tapi sebenarnya nitrogens. Okay, so that's the disadvantages of visual diagnosis. Plant analysis is very accurate. The advantage of plant analysis is it is very accurate. What is plant analysis? We will process the plant samples. Okay, for example, you have a certain plant. For example, you work as an agronomist dekat satu syarikat peladangan, kelapa sawit sebagai contoh. Lepas tu, dalam satu kawasan tu, you observe the plant like have a nutrients deficiency symptoms. It grow retarded, their yields very low, and the leaf show chlorosis symptoms. But you do not know what exactly elements involved. So you take the samples. Okay, so nak ambil samples pun ada cara dia. Kalau awak ambil pengkhususan soil plant and environments, you will learn how to samples plant part to diagnose whether the plant experience or suffer from nutrient deficiency. So biasanya kita ambil daripada daun. Okay, so daun pun ada bahagian-bahagian tertentu. Sebagai contoh, pokok kelapa sawit kita ada daun yang muda, kita ada daun yang matang, dan kita ada daun yang tua. Okay, so generally plant like that. They have young organs, the mature organs, and senescence organs. So to conduct plant analysis, we must take from mature organs. Okay, don't take from young organs or senescence organs, because we know that certain nutrients can be translocated from older organs to new organs. So that will be inaccurate if you take plant sample from a young organs or senescence organs. So usually we take from mature organs. And then we will digest. We will do digestions. What mean digestions? We will because the plant sample is a solid. Okay, to undergo nutrients analysis, you must convert the plant sample into a solutions. How we can convert a solid to solutions? By digestion process. We will convert the plant samples into solutions, and then we analyze the concentrations of certain element using, if you want to determine the concentrations of nitrogens, we can use auto analyzer. Okay? But first, the sample must be processed through JEDAL method. Okay, and then we will analyze the concentrations of nitrogens using auto analyzer. If the elements are metal elements like calciums, potassiums, apa lagi ions, we can analyze this element using atomic absorption spectrophotometer, or we can use another instruments called inductively coupled plasma optical emissions spectrometer. Okay, there are a lot of instruments that can be used for plant analysis. So we know that the amount of nitrogen is usually from 1 to 2%, and phosphorus, potassiums from 0.5% to 1%. So if the ratio of these elements lower than that percentage, we know that the plant experience this kind of nutrients deficiency. So that if you do plant analysis, we can determine accurately what kind of element involved in plant nutrients deficiency. Apart from that, if you still, we have, saya ada tunjukkan awak graph. Itu kalau pokok tu menunjukkan simptom-simptom yang jelas, okay? But we have if you have a situations where the plant did not produce observable deficient symptoms. Okay, apa maksud dia? Plant tu dia tak tumbuh, dia punya growth not comparable with a normal plant, and then their yield also not very optimum. But the plant doesn't show any deficient symptoms. So kalau kita buat plant analysis juga, kita tak dapat gambaran yang most accurate analysis. Okay, so to support, awak boleh buat juga visual diagnosis, plant analysis, and at the same times to validate our analysis earlier, okay, kita boleh juga buat soil analysis, especially kalau pokok yang berada pada hidden hunger. Okay, sebab pokok kalau dalam keadaan hidden hunger ni, symptoms tak jelas. Okay, so kita boleh buat soil analysis. Soil analysis and plant analysis, even though we determine the concentrations of element in our samples, but we need a different method. Why? Sebab apa? Because we know that if the plant tidak absorbs nitrogen, phosphorus, atau potassiums, these elements will be converted, will be assimilated into plant tissues. So they will exist in different kind of compounds. So in plant analysis, if we want to analyze the elements, we will do total element concentrations. Okay, so ingat dia punya nanti kita ada eksperimen, ada lab pasal benda ni. Okay, so kena ingat kalau untuk plant analysis we will determine total nutrient concentrations, but for soil analysis we will not determine total nutrients concentrations. If you conduct soil analysis and you determine total nutrients concentrations, you will misdiagnose the nutrients deficiency in soil. Sebab apa? You must remember, element can exist in soil in different form, okay, in different physicochemical properties. For example, phosphorus can exist in soil in the form of iron phosphate, calcium phosphates, aluminum phosphates, or it can bind with organic matter in soil. Okay, if the phosphorus bind with ions, calciums, or ions, that kind of phosphorus will not available for plant absorptions. Okay, plant tak boleh absorb phosphorus dalam bentuk tu. Plant hanya boleh absorb phosphorus is the phosphorus exists in soil in the form of phosphate. Okay, orthophosphate. There are two different kind of orthophosphate. Okay, so for soil analysis we must conduct available nutrients analysis. Okay, dekat sampel tumbuhan kita buat total concentrations elements, dekat soil kita buat available concentrations elements. Okay, so you must remember that, jangan awak tersilap nanti. Itu antara diagnosis yang that you can do to determine the deficiency symptom in plants. Apart from that, we can also conduct biochemical test. Okay, tapi jarang kita buat biochemical test. Biasa test yang kita guna adalah visual diagnosis, plant analysis, soil analysis. Okay, but sometimes certain conditions biochemical test is required to determine if the plant really suffer with specific nutrients deficiency. Okay, so saya bagi contoh for biochemical test. Kita boleh determine the concentrations of chlorophylls, okay, using spectrophotometer. You can extract the chlorophylls, okay, and then we determine using spectrophotometer. Because sometimes kita tengok daun dia berwarna kuning, okay, pokok ni with pokok biasanya contoh kelapa sawit lah, kita tengok mature oil palms will produce a green healthy leaf, okay, but you observe there are certain plant produce yellowing leaf, so you want to determine whether this plant experience from chlorosis or not, so we can conduct chlorophylls concentrations test. Okay, so if you find the concentrations of chlorophyll are significantly lower compared to normal plants, so we can conclude that plants suffer from chlorosis. So it must be nitrogen, magnesiums, or ions involved in the deficiency symptoms. Okay, faham tak setakat ni? Okay, so awak kena ingat ni lah, part ni saja. Okay, just nanti kita ada lab untuk plant analysis dengan soil analysis, so nanti awak lebih faham. Okay, kita pun ada lab untuk visual diagnosis. Okay, next slide is kenapa tak bergerak tak bergerak pula. Oh, ada banyak lagi ya. So this is nutrients deficiency symptoms that you can observe visually. Okay, so this is a general. Nanti kalau minggu ni kita akan discuss topik primary macronutrients, nitrogen, phosphorus dengan potassium. So kita akan tengok symptoms dia. Kalau plant ni suffer from nitrogen deficiency symptoms, deficiency, kita tengok symptoms dia apa. Okay, kalau the plants suffer from potassiums, what are the observable symptoms on the plants? But this is general symptoms that can be observed in plants. Why you have to know the general symptoms? Because sometimes, for example, necrosis. Okay, the necrosis can be due to nutrients deficiency symptoms. It can due to nutrients toxicity, and necrosis in plant can also be due to pathogen attacks. Okay, so sebab itulah saya cakap tadi visual diagnosis dia ada kelemahan. Kelemahan pertama dia kadang-kadang elemen yang berbeza dia ada deficit symptoms yang sama. Okay, itu dia punya kelemahan yang pertama. Kelemahan yang kedua, kita tengok the plant the necrosis. Okay, so we expect the plant would suffer from aluminum toxicity, nitrogen toxicity, dia ada deficit symptoms on certain nutrients. Okay, tapi bila kita buat chemical analysis, plant analysis, nutrients dia okay, tapi bila kita check balik sebenarnya dia attacked by pathogens. Okay, so that's why visual diagnosis dia ada kelemahan. Okay, deficit symptoms yang biasa kita tengok ataupun yang perlu kita lihat dulu adalah chlorosis. Okay, what is chlorosis? Chlorosis is yellowing. Okay, yellowing of plant leaf. Why plant leaf become yellow? Because it contains significantly lower concentrations of chlorophylls. Okay, pengalaman saya conduct lab untuk plant nutrition ada kelakar jugalah. Sebab apa? Daun pokok ni tak semestinya warna kuning. Tak semestinya warna kuning. Daun pokok ni tak semestinya warna hijau. Okay, macam pokok sawit, pokok pisang, pokok rambutan, kita tahulah daun dia warna hijau kan. Tapi ada certain pokok ni yang daunnya memang warna kuning. Okay, sebagai contoh daun bunga puding, daun bunga, certain species of flower they produce leaf with a yellow color. Okay, so bila saya buat check lab report tu, student tunjukkan gambar pokok bunga puding, which is we know that the healthy pokok puding tu memang dia warna kuning, tapi students cakap the plant experience suffer from nitrogen deficiency because the leaf color is yellow. Okay, so you have to know the normals, okay, the healthy leaf of that plant warna apa. Okay, barulah awak tengok kalau dia warna kuning, kita boleh conclude dia ada chlorosis. Tapi kalau pokok tu dah memang daun dia warna kuning, tak bolehlah kita cakap dia ada chlorosis. Okay, because we know that chlorophylls, dia ada macam-macam jenis chlorophylls kan. Chlorophylls A, chlorophylls B, and then on the plant leaf they contains a lot of pigments. Okay, anthocyanins, apa lagi? Anthocyanins biasanya warna ungu, warna kuning. Okay, depend on pH. Kalau warna biru atau ungu biasanya alkaline, but when a plant exposed to acidic, anthocyanins will give pinks ataupun yellow. Okay, so awak kena tahu tu jugalah. Okay, selain daripada plant nutrition, awak kena tahu plant physiology tu juga. Okay, so you can have a proper diagnosis. Apart from that, another deficiency symptom is burning. Okay, so what is burning? Severe localized yellowing or scorched appearance. Okay, so nanti saya ada gambar untuk apa itu chlorosis, burning, necrosis. Saya tunjukkan. Okay, apart from that, necrosis. Okay, so necrosis can occur in all living organisms. Okay, dekat badan kita pun kita boleh ada necrosis. Okay, especially when we attacked by pathogens. Same goes to the plant. Okay, especially the necrosis appear on leaf. Okay, what is necrosis? The death of plant tissues. Okay, apart from being attacked by pathogens, certain nutrients deficiency also will show necrosis symptoms. Okay, apa dia elemen yang terlibat nanti kita bincang lebih lanjut. Okay, stunting, okay, or stunted growth. So biasanya kita tengok ni dulu, chlorosis dengan dia punya growth. Okay, kita tengok dia punya apa yang lain-lain. Biasanya kita tengok dua ni dulu, chlorosis dengan stunting ataupun stunted growth. If plant experience these two symptoms, dia biasanya ada nutrients deficiency. Okay, stunting growth adalah stunted growth adalah decreased growth, shorter height of the affected plants. Okay, apart from that, molting also can nutrients deficiency in plant also can boleh dilihat daripada mottling symptoms. Okay, apa itu mottling symptoms? Spotted, irregular, inconsistent patterns. Okay, dia punya daun macam kerinting. Okay, ataupun dia ada bintik-bintik. Nanti saya bagi contoh. Okay, ini adalah chlorosis. Okay, so chlorosis juga ada berbagai-bagai patterns. Okay, sebagai contoh, this is the chlorosis occur in the mid veins or the plants. Okay, kalau awak tengok gambar yang pertama, the first picture and then the last picture, the chlorosis appear at even though the chlorosis appear at the leaf, but the patterns are different. The first picture, the chlorosis appear at mid vein. Okay, gambar yang ketiga ni dia dekat apa namanya, saya lupa lah punya terms. Dia mid veins dia tak ada chlorosis. Okay, tapi dekat bahagian yang lain dia ada chlorosis kan. Kalau awak nampak beza kan dengan yang pertama ni. Okay, yang ini, yang gambar tengah-tengah, the second picture, the chlorosis appear at the wool leaf. Okay, dekat semua daun ni dia ada chlorosis. Okay, dia ada, ini involve a different pattern of chlorosis, involve different kind of elements. Okay, kalau tak silap saya ini adalah magnesium. Okay, yang ini adalah iron. Okay, yang ini saya tak tahu apa. Tapi kalau ini nitrogen magnesium. Kalau ions dia dekat bahagian yang daun baru nak terbentuk, bahagian daun muda. Okay, this is necrosis. Okay, necrosis adalah daun tu mati. Okay, the spotted they produce spotted tissue death. If the plant attacked by virus, bacteria or pathogenic fungus, the same symptoms can also appear. Okay, so necrosis adalah satu contoh bagaimana tumbuhan nak mengelakkan tisu-tisu that being attacked by pathogens daripada tersebar, pathogen tersebar ke tisu yang lain. So dia bunuh diri. Okay, tapi untuk deficiency symptoms, kita boleh juga tengok ada necrosis. Okay, sebab apa? Biasanya ini adalah apa ya? Kalsium kalau tak silap saya. Okay, so sebab apa? Tisu-tisu ni dah tak boleh berfungsi sebab kekurangan elemen tu tadi. Okay, awak kena ingat salah satu karakteristik elemen apa? A plant cannot continue its life cycles. Okay, if this element dia tak hadir. Okay, elemen ni hadir tapi dalam jumlah yang kurang. Okay, tapi certain tisu tu elemen tu tak ada. So apa terjadi? Tisu tu mati. Okay, ini adalah mottling, dia berbintik-bintik. Okay, so this is mottling at daun cili. Okay, yang ini adalah daun anggur. Okay, dia macam chlorosis. Tapi chlorosis dia tak mati. Yang ini dia macam necrosis and also chlorosis. Okay, sebab kalau chlorosis dia kuning saja. Kalau chlorosis, necrosis dia mati. Tapi dia macam mix between necrosis and chlorosis. Jadi ada warna kuning. Lepas tu kalau severe dia akan mati. Bahagian-bahagian ni dia akan mati. Okay, this is leaf burning. Okay, at the edge of a leaf, the tissue dying. Okay, dia macam necrosis tapi kita panggil leaf burning. Okay, dekat hujung dia. So ini adalah simptom-simptom kalsium. Okay, so kekurangan kalsium dia tak dapat kalsium penting untuk osmosis. Okay? So dia tak ada kalsium dekat sini, daun tisu tu mati. Okay, calcium is also important for calcium peptide. Okay, dia untuk joining between the adjacent cells dekat daun atau sel tisu tumbuhan. Any questions? So first lecture saya bagi general saja. Okay, setiap topik-topik yang we discuss in first lecture, we will discuss more details in every week. Okay. So any questions before we continue with our second topic? Second lecture. Any questions?
No, doktor.
No.
Tak ada no questions ya. No questions, either you fully understands or awak kadang-kadang tak tahu tapi takut nak tanya. Okay. If you have query or things you didn't understand, awak just tanya saja. Okay. Kita take a rest lah sekejap before we continue. Insyirah.
Okay, doktor.
Okay, doktor.
Okay. Okay, 3 suku kita sambung semula. Okay.